Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test question paper for the subject Plant Biotechnology conducted in the year 30th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Plant Biotechnology, you have to go to my channel Omni Bio Entrance, search for playlist. Then in the playlist you search for CUET and then you will get the previous year question papers. Coming to the first question, which of the following is a growth stimulant stimulating shoot growth under standard tissue culture conditions? So it is benzyl aminopurine, it is a cytokinin, whereas TIBA is an auxin, abscisic acid and CUET are not cytokinin, so BAP is the answer. Then next one, which of the following is not a necessary facility for standard tissue culture laboratory? So it does not require thermocycles, whereas other options, that is a media preparation room, growth room with controlled condition, autoclave, all these are required. Thermocycles is seen in PCR. Then next one. Chromosome elimination is most commonly used for the production of haploids in which group of plants? So it is seen in cereals like wheat and barley. Answer is option 1. Then which of the following is a method of indirect gene transfer in plants? So indirect gene transfer, it involves the use of vectors to deliver genetic material into the plant cell. So here out of these options, microprojectile, microinjection, electroporation does not use vectors, whereas agrobacterium mediated gene transfer uses vectors. So the answer here is option 3. Then next one. Successful production of haploids from anther culture in Datura was reported by. So it was reported by Guha and Maheshwari. Answer is option 1. Then assertion reason question. Assertion. The utility of haploids in plant breeding eventually requires production of double haploids. That is correct, because haploids are sterile and they have genetic instability and limited seed production. So you require the haploids being converted to double haploids and then only used in plant breeding. Reason what they have given is double haploids may be produced spontaneously or by treatment with colchicine. They have given the reason as how double haploids are produced. They have not given the reason as why double haploids are required. So the answer here is both A and R are correct, but R is not the correct explanation of A. Then next one. Terminal differentiation of bacteroids is controlled by the production of nodule-specific cysteine-rich peptides. That's correct. Bacteroids can neither divide nor revert back to the non-differential state. Yes, that is the reason for A. So both A and R are correct, and R is the correct explanation of A. Then next one. Stages in micropropagation. So you have to choose with the correct sequence. So first will be choice of the explant. D. You have to choose the explant, whether it is a leaf or a shoot or stem, you have to choose. So choice of explant. Then comes surface sterilization of the explant material. You have to go for surface sterilization if it is from an outside grown, that is not in tissue culture bottle. If it is grown outside in the soil under normal atmospheric conditions, you have to surface sterilize the explant. Then establishment of tissue in vitro. Then you have to establish the tissue in vitro conditions. Then multiplication of shoots and or roots. Then this explant will give rise to multiple shoots or roots. And acclimatization and hardening. Finally, it will be acclimatization and hardening. You have to transfer the plant outside, that is hardening. So the option will be DACEB. Answer is option 2. Then next one. Growth curve of bacteria in a batch culture consists of phases. So growth curve is having lag phase, then log phase or exponential phase, then comes stationary phase and finally death phase. But here there is one more option, long-term stationary phase, that is after the death phase. So here the sequence will be lag phase. C will be the beginning, then comes E, that is log phase. So 3 is the answer. Then B, that is stationary phase, and then finally death phase and long-term stationary phase. So the answer here is option 3. Then next one. When in an interspecific or intergeneric hybrid, the chromosomes of one parental genome are preferentially eliminated, it is called chromosome elimination. We just read that in case of cereals and barley, chromosome elimination is used. That was one of the previous questions. Then chromosome elimination is widely used in the production of haploids in wheat and barley. So statement 2 is also correct. So both statements 1 and statement 2 are correct. Then composition of culture medium. Which of the following statement is true? The culture media need not be sterilized. Culture media has to be sterilized before culturing. So A is not correct. The major constituents of culture medium include inorganic nutrients, organic nutrients and growth hormones. Yes, that is correct. B is correct. The same nutrient media can be used effectively for all plants. No, because different plant species have different nutrient requirements. Some require more copper, some require more manganese. Then different plants also need different hormonal conditions. So C is not correct. The various culture media differ mainly in the quantity rather than quality of the nutrient components. That is also correct. So B and D is the answer. Answer is option 4. Then next one. Neomycin phosphotransferase. Selectable marker gene or reporter gene and substrate used for selection. So neomycin phosphotransferase uses kanamycin. A4. Hygromycin phosphotransferase uses hygromycin. B1. Beta-glucuronidase uses X-gluc. C2. And galactosidase uses X-gal. D3. So the answer here is option 4. Then next one, who gave the concept of cellular totipotency? So it was G. Haberland who gave the concept of cellular totipotency. Then cross between two general species or varieties which cannot be hybridized using conventional method is called. So it is called wide hybridization. Now there is another option, that is somatic hybridization. Somatic hybridization is a technique used in wide hybridization. So the correct answer will be wide hybridization. Then next one, function and spatial distribution. Oxygen protection. Oxygen protection is for nitrogenase enzyme. So it will be inside the root nodules. A4. A4 is there only in option 2. So option 2 must be the answer. Then nitrogen fixation, that is bacterial cytoplasm, ammonia production, plant cytoplasm, N2O production, release in the atmosphere. D3. So answer here is A4, B1, C2 and D3. Then next one. bacteria and their characteristics. Corynebacterium. Corynebacterium is club-shaped cells. A3. A3 is there only in option 2, so that must be the answer. Then mycobacteria, they are acid fast. Nocardia, they are rudimentary branching. And thermoactinomycetes, they are high temperature growth. So answer here is option 2, A3, B1, C4 and D2. Then next one, advantage of synthetic seeds are. So synthetic seeds, they are known as artificial seeds or they can be also called as somatic seeds, and they are encapsulated plant embryos, and they are produced mainly through tissue culture, and these can germinate and grow into new plant. So advantages of synthetic seeds. They can be stored for up to a year without loss of viability. That is correct. They need special structures like greenhouses for hardening and establishment. Need not. They are easy to transport. That is also correct. They can be directly sown into soil like natural seeds. That is also correct. So A, C, D is the answer. Answer is option 2. Then next one. Hybrids can be obtained using one of the following methods. So hybrids, they are called as cytoplasmic hybrids, and they combine the cytoplasm of one cell with the nucleus of an other cell. That is how hybrids are formed. So one cell is having the nucleus, whereas the other is not having the nucleus, but it is providing the cytoplasm. So hybrids are elimination of any one parent cell. That's not correct. You don't eliminate any parent cell. Antibiotic resistance in emulated cell. That is also not correct. Fusion of protoplast from normal parent and non-viable nuclei from other cell. Yes, that is how hybrids are produced. So protoplast will be from the normal parent, and the other protoplast will be from the, or another will be either an enucleated protoplast, or it will be having non-viable nuclei. You fuse both of them and you get a hybrid. So C is correct. Then fusion of normal protoplast with enucleated protoplast. That is also correct. Selective elimination of one of the nuclei from the heterokaryon. Heterokaryon is the product that you get after fusion of two nuclei. So what you do is, after heterokaryon production, you selectively eliminate one of the nuclei. So the answer here is C, D and E. Answer is option 2. Then next one. In some crops, especially ornamentals, it is useful to produce virus-free plants at commercial level. That's correct. These plants can be obtained via explants obtained from virus-free plants or through apical meristems, which are generally free of infection. That's also correct. So both statements 1 and 2 are correct. Answer is option 1. Then next one. Pollen grains of many crop species exhibit pollen dimorphism. Now what is pollen dimorphism? It is the presence of two distinct forms. That is two distinct morphological forms of pollen grains within a single plant species. That is called as pollen dimorphism. And dimorphism, it can be seen in the size, shape or surface ornamentation of the pollen. So next statement, in the crop species that exhibit pollen dimorphism, it is the small grain, S grain, that respond during anther culture. So here that statement also is correct. So both statements 1 and 2 are correct. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of plant biotechnology being answered, you can go to my channel Omni Bio Entrance. In the playlist, search for CUET. You will get there. Thank you.